Specialty Grand Challenge article. Meetings on visual Specialty Grand Challenge article. Front. Bioinform June 17, 2021. Secretary Data Visualization. Volume 1 to 2021. https://doi.org/10.3389/fnf.2021.669186. Grand Challenges in Bioinformatics Data Visualization. www.frontierson.org. Sean I. O''Donahue 1,2,3. 1 Garvan Institute of Medical Research, Darlinghurst, NSW, Australia. 2 School of Biotechnology and Biomolecular Sciences, University of New South Wales, Kensington, NSW, Australia. 3.0 Data 61, Eveleigh, NSW, Australia. Introduction. Increasingly, the life sciences rely on data science, an emerging discipline in which visualization plays a critical role. Visualization is particularly important with challenging data from cutting-edge experimental techniques, such as 3D genomics, spatial transcriptomics, 3D proteomics, epiproteomics, high-throughput imaging, and metagenomics. Data visualization also plays an increasing role in how research is communicated. Some scientists still think of data visualization as optional. However, as more realize it is an essential tool for revealing insights buried in complex data, bioinformatics visualization is emerging as a subdiscipline. This article outlines current and future grand challenges in bioinformatics data visualization and announces the first publication venue dedicated to this subdiscipline. Over the past 2 decades, life science data have increased rapidly in volume and complexity, with the result that data analysis is often the major bottleneck. O'Donoghue et al., 2010a. For example, all major genomics breakthroughs so far have been accompanied by the development of groundbreaking statistical and computational methods, Green et al., 2020. Thus, in the remaining decades of the 21st century, life scientists will become increasingly reliant on the emerging tools and methods of data science, Bly and Smith, 2017, Altman and Levitt, 2018. One of these methods is data visualization, aka data vis, which plays a critical role in transforming data and analysis outcomes into insight. Card et al., 1999. Data visualization involves analysis, design, and rendering, as well as observation and cognitive processing. Figure 1. Some scientists think of data vis as an optional step mostly aimed at aesthetics. However, there is growing recognition that it is an essential tool in the analysis of complex data. 2 indicators of this recognition are the recent sales of data vis companies Looker and Tableau for UseDeb and $16 billion respectively. Figure 1. www.frontierson.org. Figure 1. The data science cycle. Analysis of newly acquired data increasingly relies on integration with large accumulating volumes of complex pre-existing data and requires frequent reanalysis and re-rendering. Visualization is the main way researchers observe both raw and analyzed data. An overarching grand challenge of data visualization is to leverage human visual capabilities, which involve most of the brain and can process 10 million bits per second, Koch et al., 2006, to recognize patterns within 100 milliseconds, Healy and Enns, 2012, thereby transforming data into insight. These insights in turn lead to new hypotheses, thus continuing the cycle. Unfortunately, the critical step of manually validating derived models by visually comparing raw versus analyzed data, Anscombe 1973, is often overlooked. Currently, however, most attention is focused on another aspect of data science, namely, the use of machine learning to develop artificial intelligence systems. Such systems have recently led to exciting advances in the life sciences, for example, Callaway, 2020a, but also to some hyperbole. Clearly, machine learning methods are increasingly critical for research, but these methods also have limitations. Challen et al., 2019, Heaven 2019, Yu and Kahan 2019. More fundamentally, automated methods are insufficient, since analysis outcomes must be observed and understood by an analyst before insight can occur. Figure 1. Most analysts use data visualization as an integral part of their cognitive processes. Especially important is manual validation, which involves checking for errors and outliers in raw data, and for wrong assumptions used in automated analysis methods. Anscombe, 1973. Automated data analysis, including machine learning, and data visualization are just components of the larger goal of data science, which the eminent computer scientist Fred Brooks argues should focus on intelligence amplification, aka IA, i.e., on amplifying our abilities to manage more complex data. Brooks, 1996. In my opinion, helping achieve the goal of IA is the overarching grand challenge of data vis. Prioritizing grand challenges in bioinformatics. Since data visualization aims to amplify human intelligence, we could ask ourselves one, of all our colleagues working across different life sciences, whose intelligence most needs amplifying? Or, humor aside, which fields are creating data that are both important and urgently need improvements in visual analysis? Addressing this question is the core mission of an annual series of international meetings on visualizing biological data, VISB2. From my perspective as chair of this meeting series, it is clear that the biological and biomedical sciences are currently awash with vexing data challenges where current analysis methods and tools are fundamentally inadequate. Thus, researchers looking for grand challenges in bioinformatics data visualization, aka BioVis, are spoiled for choice. Of very many worthy challenges, below are 6 that have been highlighted repeatedly by VISB speakers over the past decade, as cases in which innovations in visual analysis are likely to lead to significant breakthroughs in our understanding of life. Figure 2 showcases some of the visual methods currently being used to partly address these challenges. This list of challenges is far from comprehensive. Researchers already focused in a particular field of the life sciences, for example drug design, medicine, ecology, would likely prioritize very different lists of worthy challenges. Figure 2. www.frontierson.org. Figure 2. 6 data visualization methods currently used in bioinformatics grand challenges. A molecular scale 3D model of the human genome may soon be feasible. In preparation, visualization tools are being developed to enable interactive, multi-scale exploration of such models, adapted from Asbury et al., 2010. B. TSNE analysis of Skriner-Sek data on breast cancer metastases, adapted from Valdez-Moore et al., 2019. C. Spatiotemporal graph of phosphorylation events in fat cells following insulin stimulation, adapted from Ma et al., 2015 by Jenny Vuong. D. Here, an interactive, web-based data integration environment is used to explore and curate a molecular-scale model of a subcellular landscape showing HIV host interactions, Klein et al., 2018, Oten et al., 2020. E. Portion of spatial transcriptomics analysis showing expression of 2 genes in an anterior slice from mouse brain, adapted from Vandenbaan and Diess, 2020. F. Disease trajectory graph showing progression from heart pain to cardiac arrest across the entire Danish population, adapted from Jensen et al., 2014. I. In genomics, there is rapid progress towards the goal of determining the spatiotemporal organization of chromosomes at molecular scale resolution, Figure 2a. This is driven by advances in sequencing technologies that can infer spatial contacts, Lieberman-Aden et al., 2009, as well as in high-resolution imaging, et al., 2017. Integrating these multi-scale and multimodal data poses formidable visualization challenges. And Noble, 2015, Sarah et al., 2015. However, achieving this goal would transform our understanding of what gets transcribed and how and when transcription is controlled in different cell types. 2. In transcriptomics, rapid advances in single-cell RNA-seq, Skriner-Sek techniques now make it possible to track behaviors of individual cells in unprecedented detail, providing a window into events that were previously hidden, Wills et al., 2013. For example, Skriner-Sek can be used to track cell differentiation and the evolution of cell-cell contacts during the growth of cancerous tumors, for example, Valdez-Moore et al., 2019. Also, in combination with imaging methods, these techniques can be used to resolve the spatial location of RNA transcripts within single cells, Chen et al., 2015. Each type of scRNA-seq experiment produces data that are both highly multidimensional but also very specific. Tailoring effective data analysis strategies for each type of experiment requires development of innovative visual analysis methods to overcome limitations with existing generic approaches such as T-SNE distributed stochastic neighbor embedding, Figure 2b, van der Motten and Hinton, 2008, or diffusion maps, Quaffman et al., 2005. This challenge currently engages many bioinformaticians, driven by the promise of discovering the key mechanisms used to control cellular processes. 3. In proteomics, advances in high-throughput mass spectroscopy, Kim et al., 2006, Morel et al., 2006, Olson et al., 2006, have begun to provide first glimpses into the highly dynamic epiproteome, i.e., the set of all post-translational modifications, PTMs, made to all proteins in a cell, Jung et al., 2016, Kaur et al., 2019. So far, at least 200 distinct types of PTMs are known, and related advances are revealing that comparable levels of complexity occur in modifications seen to both RNA, Rountree et al., 2017, and lipids, Shevchenko and Simons, 2010. Currently, most of these modifications are poorly studied. Even phosphorylation of human proteins, one of the best studied PTMs, gives rise to a phosphoproteome that is still largely unknown or dark. Needham et al., 2019. However, this is set to change rapidly over the next few years, although extracting insights from the dynamic, highly multidimensional datasets from epiproteomics, Figure 2c, epitranscriptomics, and lipidomics remains a major challenge, Kaur et al., 2019, Kaur et al., 2020. Nonetheless, the insights gained are likely to fundamentally advance our understanding of cellular processes in health and diseases, for example, by revealing molecular events that occur during illness or following therapeutic interventions. 4. In cell biology, a convergence of several experimental techniques and computational methods are driving work towards an audacious goal, determining the spatiotemporal organization of a human cell at molecular resolution, Tomita, 2001, Singla et al., 2018. The spatial location of proteins can be mapped at subcellular resolution using imaging and mass spectrometry techniques, Boisvert et al., 2012, Gatto et al., 2019, Lundberg and Borner, 2019. The molecular structure of these proteins can be determined using cryogenic electron microscopy, Bay et al., 2015, Callaway, 2020b, even when they occur in large complexes. Transient protein complexes can be either measured experimentally, inferred from sequence information, Ellefson, 2021, or modeled in large-scale molecular simulations, for example, McGuffey and Ilcock, 2010, Fag et al., 2015. Still largely unmet, Figure 2d, is the formidable challenge of developing visual methods that integrate these data with information on protein-protein interactions, Glenborg et al., 2010, Ghosh et al., 2011, Protein small molecule interactions, Krohn et al., 2016, Protein 3D structure, O'Donoghue et al., 2010b, Johnson et al., 2015, Kozlakova et al., 2017, Olson, 2018, and Protein dynamics, Humphrey et al., 1996, Rasavi et al., 2014, Farina and Daggett, 2019. If this challenge can be met, this would provide a structural framework for understanding the molecular basis of cell behavior. This in turn could have profound impact, similar to how the structure of DNA advanced our understanding of the molecular basis of information storage and replication. Watson and Crick, 1953. V. Multiple advances in tissue-scale imaging are driving other audacious goals. For example, two-photon fluorescence microscopy, Pittet and Wieselder, 2011, is being used to construct 3D maps of neural connectivity in mammalian brains, for example, Economo et al., 2016, and also to track real-time movements of cells in subcellular structures within living tissues, including tumors, for example, Kedron et al., 2008, Conway et al., 2018. In addition, combining tissue imaging with fluorescence in situ hybridization methods now enables spatial mapping of RNA transcription, Stahl et al., 2016, Burgess, 2019, at near cellular resolution, Figure 2e, Stickles et al., 2020, Marks, 2021. Combining these data with tissue scale or whole-body kinetic modeling, Al-Qahtani 2017, has potential to revolutionize our understanding of physiology and the body's responses to events such as tumor growth or therapeutic interventions. However, extracting insight from such massive, complex datasets requires development of highly tailored, innovative visual analysis methods. For example, Santos et al. 2015, GTEx Consortium 2017, Ullin et al. 2017, to address the many challenges of bridging molecular information with tissue and whole organism scale data. Walter et al. 2010, O'Donoghue et al. 2018. Finally, a set of daunting challenges lie in comparing temporal changes in clinical records across cohorts, Kerchevsky and Snyder, 2018. The complexity and volume of these data are increasing rapidly due to wearable devices, Kim et al. 2019, Ray et al. 2019. However, data dimensionality dramatically increases when microbiome analysis is also included. For example, Schussler-Fiorenzo-Rose et al. 2019. Current visual analysis methods are often inadequate, even when exploring the microbiome of a single person, Proctor et al. 2010, Pasoli et al. 2019. Addressing these many challenges calls for innovative new approaches in how we visualize phylogenetic, for example Rosendahl and Harmon 2012, Letunik and Bork 2019, and pangenomic relationships, for example Ding et al. 2018, how we compare microbiomes, for example Caparaso et al. 2010, Darling 2004, Waterhouse et al. 2009, and how we explore clinical information gathered from large cohorts, Figure 2F, for example Jensen et al. 2014. Communicating science visually. Once any of the above grand challenges are addressed, a new challenge is created: how to convey the significance of this breakthrough to others. Science isn't complete until it's communicated. Day, 1998. But the highly specific nature of the life sciences can make it difficult to communicate a breakthrough, even to researchers working in closely related fields, let alone to the general public. Here again, data visualization plays an increasingly central role. Many of the visualization methods and tools designed for analysis can be repurposed for communication, but often dedicated communication approaches need to be developed to address specific data challenges, especially when conveying complex or unfamiliar ideas. For example, an intrinsic difficulty with communicating insights from a molecular scale model of a human cell, challenge 4 above, is that mesoscale molecules, Johnson et al. 2015, Goodsell et al. 2018, behave very differently to macroscopic objects. This difficulty is driving development of innovative communication approaches to convey these dynamic behaviors, for example, via 2D illustration, for example, Gardner et al. 2018, or 3D graphics, Goodsell et al. 2020, for example, Music et al. 2015, Walden et al. 2019. In turn, such methods are being used to create informative and inspiring videos. 4. McGill, 2008, Johnson and Herdig, 2014, Iwasa, 2015, and to build interactive environments that can be explored with virtual reality techniques, Johnston et al., 2018. In contrast to visual analysis, subjective qualities such as aesthetics and novelty become important when using visual methods for outreach. However, the impact of visual storytelling goes beyond outreach. The difficult process of assembling our hypotheses into clear visual narratives, Nyack et al., 2020, invariably involves integrating pre-existing data in new ways, often revealing hidden assumptions and knowledge gaps. This, in turn, often leads to new insights and hypotheses, for example, Riley and Ingber, 2017, thereby continuing the data science cycle, Figure 1. Thus, visual communication should also be considered as an intrinsic part of any grand challenge in bioinformatics data visualization. Bridging bioinformatics and visualization research. Addressing the above grand challenges requires combining expertise in visual analysis with specific knowledge about the biological context of each experiment and about what can be inferred, given expected errors and given prior knowledge. This, in turn, requires an exchange of knowledge between researchers in computer science and in various life sciences. Unfortunately, these communities rarely attend the same meetings, have very different publication practices, and are strongly disincentivized to collaborate, since their work performance and funding are assessed using fundamentally different metrics. To help counteract these obstacles, a range of resources have been created for life scientists that showcase how data visualization is transforming biology. These include special issues of Nature Methods 5, Eveleigh 2010, and of the Journal of Molecular Biology 6, a section of BMC Bioinformatics dedicated to advances in either data visualization or image analysis 7, a Nature Methods article series on visualization issues 8, and the VISB 9 meeting series mentioned above, which was highlighted in Nature News, Callaway 2016. Corresponding resources have also been created for computer scientists, including several dogstool reports, Gorg et al. 2013, Ertz et al. 2018, and regular meetings, including the VCBM 10, Visual Computing for Biology and Medicine, and BioVis 11 workshops, co-located with the annual conferences, Eurographics 12, and ISMB 13/IEEE Vis 14, respectively. In addition, other international meetings bridge related communities, but with more targeted focus. For example, Biomedical Data Visualization, Holzinger 2012, O'Donoghue et al. 2018, is the focus of MediVis 15, while Molecular Graphics, Olson 2018, Martinez et al. 2019, is the focus of Malva 16 and of several Shonen meetings, Schafferhans et al. 2016, Bodden et al. 2018. These activities have catalyzed adoption of emerging visualization methods in the life sciences. Notable examples include use of ggplot2, Wickham 2009, for offline analysis, use of D3, Bostic et al. 2011, for interactive data exploration in websites, use of virtual reality in molecular graphics, Music et al. 2015, Summer et al. 2018, use of augmented reality in surgery, Bernhardt et al. 2017, Meyer-Hein et al. 2017, and the use of interactive volume rendering for full-body virtual autopsies, Jung et al. 2006. Looking forward, advances in computer hardware and software are set to provide greatly improved graphics as well as new paradigms for user interaction. One of the overarching grand challenges in BioVis is to use these advances to improve research, communication, training, and clinical practices. Publishing advances in bioinformatics data visualization. As with all frontier interdisciplinary work, publishing advances in BioVis can be problematic. Publication venues in computer science often reject manuscripts describing novel methods that lack broad applicability, or describing novel tools that lack a user study, even when the advances described are obviously of very high value to domain experts. Life science journals often reject manuscripts describing novel visual analysis approaches that are too technical or have not yet been used to derive significant, novel biological insights, even when the advances described are highly innovative or required enormous effort. Even journals specializing in bioinformatics often reject manuscripts that describe user studies, design studies, or improvements to existing tools. Advances in BioVis could lead to tremendous impact by improving the tools used by life science researchers. However, publication decisions are often driven by perceived potential impact, a criterion that frequently rejects even the most groundbreaking work, Bjork 2020. To address this issue, several open-access publishers such as Frontiers 17, BMC 18, and Peerage 19 have emerged in the past decades with the mandate to base publication decisions solely on scientific rigor and reproducibility. As part of this process, this Grand Challenge article has been written to accompany the launch of the data visualization section in the newly created journal, Frontiers in Bioinformatics, the first publication venue dedicated to bioinformatics data visualization. By collecting advances across all life sciences, the data visualization section will facilitate exchange of knowledge and best practices between research groups that may otherwise never cross paths. Mostly, these groups will comprise bioinformaticians, biomedical researchers, computer scientists, and science communicators, but BioVis also engages educators, user experience designers, as well as visual arts practitioners, particularly graphic designers and medical illustrators. Conclusion. It is fortunate that bioinformatics data visualization engages a broad community with diverse backgrounds and perspectives, since one of our core processes is to overcome current cognitive biases in analysis and to find more effective ways of seeing, analyzing, and thinking about our data. A historical exemplar of this process is the inspiring interdisciplinary work of Jane and David Richardson 20, who devised a method for transforming complex, all-atom representations of large protein structural models into ribbon representations that are greatly simplified and often insightful. Richardson, 1981. Richardson and Richardson, 1989. Our task going forward is to find analogous ways to reimagine the much larger and more complex datasets in today's grand challenges, and, using current and future advances in computer graphics, to invent simplifying transformations that are also insightful. Each such invention can be thought of as a step in creating a new visual language, Lima, 2011, that will enhance how we explore, describe, and communicate the processes of life. Given the daunting data challenges already at hand, creating this visual language will likely be difficult and will require considerable creativity combined with statistical and mathematical rigor. Given the even more daunting data challenges that are set to come, we could say that bioinformatics data visualization has barely begun. O'Donoghue et al., 2010a. Author contributions. Syed was the sole author of this article. Conflict of interest. The author declares that the research was conducted in the absence of any commercial or financial relationships that could be construed as a potential conflict of interest. Acknowledgements. The perspectives presented in this article have emerged from discussions with participants of the 10 VISB meetings to date. I would like to thank my VISB co-chair James Proctor, as well as Andrea Schafferhans, Nablina Sikta, and Sandeep Kaur for helpful discussions and feedback. Finally, thanks to Wikimedia user Hankum for creating the brain illustration used as part of Figure 1, and for releasing this illustration into the public domain. Footnotes. 1. Paraphrasing from Fred Brooks, 1996. 2. https://visb.org/ 3. https://www.uniprot.org/docs/tmlist 4. https://chlorophy.com/showcase/ 5. https://www.nature.com/collections/ikikii 6. https://www.sciencedirect.com/journal/Journal of Molecular Biology/special issue/10 VZQRR 6 PS7 7. https://bmcbioinformatics.biomedcentral.com/articles/sections/imaging image analysis and data visualization 8. http://blogs.nature.com/Mythagora/2013/07/data visualization points of view.html 9. https://visb.org/ 10. https://dbop.org/db/conference/vcbm 11. http://biovis.net/ 12. https://www.e.g.org/ 13. https://www.iscb.org/aboutism 14. http://evis.org/ 15. http://www.graphicslink.co.uk/medi/vis 2021/ 16. http://decibel.phi.muni.cz/expisca/malva/. 17. Frontiers does not reject manuscripts based on their perceived potential impact. https://www.frontierson.org/about/review system. 18. We do not make editorial decisions on the basis of the interest of a study or its likely impact. https://bmcbioinformatics.biomedcentral.com/about. 19 https://peerge.com/about/FAQ #quality 20 https://stories.duke.edu/sciences Mother of Ribbon Diagram celebrates 50 years at Duke. References: Ertz, J., Glenborg, N., Mari, G. E., and Nieselt, K. K. 2018. Visualization of Biological Data Crossroads, Dogstool Seming, 18161. Dogstool Rep. 84, 32-71. DOI 10.4230/dagrep.8.4.32. Cross-ref full text. Al-Qahtani, S. 2017. In Silico Ad Tox Modeling, Progress and Prospects. Expert Oppen, Drug Metab, Toxical. 13, 1147-1158. DOI 10.10801742525.2017.1389897. PubMed Abstract, Cross-Ref Full Text Google Scholar. Altman, R.B., and Levitt, M. 2018. What is biomedical data science and do we need an annual review of it? Anu, Reverend Biomed Data Sci 1, I3, DOI 10.1146/Annual BDO 104-1718-100001. Cross-Ref Full Text Google Scholar. Anscombe, F.J., 1973. Graphs in Statistical Analysis, MStat 27, 17-21. DOI 10.10800331389.104789.6610.2307 2,682,899. Cross-Ref Full Text Google Scholar. Asbury, T.M., Mittman, M., Tang, J., and Zheng, W.J., 2010. Genome 3D, a viewer model framework for integrating and visualizing multi-scale epigenomic information within a three-dimensional genome. BMC Bioinformatics 11, 444, DOI 10.1186/1471-2105-11-444. PubMed Abstract, Cross-Ref Full Text Google Scholar. Otan L., Mariton M., Barbaro B.A., Gardner A., Olson A.J., Sanner M. et al., 2020. Mesoscope, a web-based tool for mesoscale data integration and curation. Workshop Mole Graph, Vis Anal Mole, Data 9, 23-31. DOI 10.2312/Malva.20201098. Cross-Ref Full Text Google Scholar. I. F. and Noble W.S. 2015. Analysis Methods for Studying the 3D Architecture of the Genome. Genome Bio 16, 183, DOI 10.1186/S13059015075-7. PubMed Abstract, Cross-Ref Full Text Google Scholar. Bodden, M., O'Donoghue, S., and Sikijima, M., 2018. Web Molecular Graphics, Emerging Technologies and Standards. Japanese Shonen Meeting Report, number 142. Available at https://shonen.ni.ac.jp/doc/no142.pdf. Bay, X, McMullen G. and Scares SHW 2015. How Cryo-EM is Revolutionizing Structural Biology. Trends Biochem, Sci 40, 49-57. DOI 10.1016/J.Tibbs.2014.10.005. PubMed Abstract, Cross-Ref Full Text Google Scholar. Bernhardt S., Nicolau S.A., Soler L., and Doignan C., 2017. The Status of Augmented Reality in Laparoscopic Surgery as of 2016. Med Image Anal 37, 66-90. DOI 10.1016/J.Media.2017.01.07. PubMed Abstract, Cross-Ref Full Text Google Scholar. Bjork, R., 2020. The Journals in Physics that Publish Nobel Prize Research. Scientometrics 122, 817-823. DOI 10.1007/S11192019033128. Cross-ref full text, Google Scholar. Bly, D.M., and Smith, P. 2017. Science and Data Science, Proc National, ACADSci 114, 8689 to 8692, DOI 10.1073/NS.17020761114. PubMed Abstract, Cross-ref full text, Google Scholar. Boisvert, F.M., Ahmad Y., Gierlinski, M., Charrier, F., Lamont D., Scott M. et al., 2012. A quantitative spatial proteomics analysis of proteome turnover in human cells. Mole Cell Proteomics 11, M111.011429, DOI 10.1074/MCP.M111.011429. Cross-ref full text, Google Scholar. Bostock, M., Ogievetsky, V., and Hare, J., 2011. D3, Data-Driven Documents, IEEE Transvis Compute Graph 17, 2301-2309. DOI 10.1109/TVCG.2011.185. PubMed Abstract, Cross-ref full text, Google Scholar. Brooks, F.P., 1996. The Computer Scientist as Toolsmith, 2, Common ACM 39, 61-68, DOI 10.1145/227,234.227243. Cross-ref full text, Google Scholar. Burgess, D.J., 2019. Spatial Transcriptomics Coming of Age, Nat Reverend Janet 20, 317. DOI 10.1038/S4157601901129Z. PubMed Abstract, Cross-ref full text, Google Scholar. Callaway, E., 2016. The Visualizations Transforming Biology, Nature 535, 187-188. DOI 10.1038/35187A. PubMed Abstract, Cross-ref full text, Google Scholar. Callaway, E., 2020a. It will change everything. DeepMind's AI makes gigantic leap in solving protein structures. Nature 588, 203-204, DOI 10.1038/D415860200033484. Cross-ref full text, Google Scholar. Callaway, E., 2020b. Revolutionary cryo-EM is taking over structural biology. Nature 578, 201, DOI 10.1038/D4158602003419. PubMed Abstract, Cross-ref full text, Google Scholar. Caparaso, J.G., Kaczynski, J., Stambaugh, J., Bittinger, K., Bushman, F.D., Costello, E.K. et al., 2010. Quime allows analysis of high-throughput community sequencing data. NAT Method 7, 335-336. DOI 10.1038/nmeth.f.303. PubMed Abstract, Cross-ref full text, Google Scholar. Card, S.K., McKinley, J.D., and Schneiderman, B., 1999. Readings in Information Visualization, Using Vision to Think, the Morgan Kaufman Series in Interactive Technologies, San Francisco, California, Morgan Kaufman Publishers. Challen, R., Denny, J., Pitt, M., Gompels, L., Edwards, T., and Seneva Atanasova, K., 2019. Artificial Intelligence, Bias, and Clinical Safety. BMJ Qual Software 28, 231-237. DOI 10.1136 at BMJQS 2018-008-37. PubMed Abstract, Cross-ref full text, Google Scholar. Chen, K.H., Bodeger, A.N., Moffat, J.R., Wang, S., and Zhuang, X. 2015. Spatially Resolved, Highly Multiplexed RNA Profiling in Single Cells. Science 348, AAA 6090. DOI 10.1126/Science.AAA 6090. PubMed Abstract, Crossref Full Text Google Scholar. Quaffman R.R., Lofan S., Lee A.B., Maglioni M., Nadler B., Warner F. et al., 2005. Geometric Diffusions as a Tool for Harmonic Analysis and Structured Definition of Data. Diffusion Maps. Prognostic SI 102, 7426 to 7431. DOI 10.1073/INIS.05003-34102. PubMed Abstract, Crossref Full Text Google Scholar. Conway, J.R.W., Warren, S.C., Herman, D., Murphy, K.J., Kazay, A.S., Venin C. et al., 2018. Intravital imaging to monitor therapeutic response in moving hypoxic regions resistant to peak pathway targeting in pancreatic cancer. Cell Rep 23, 3312 to 3326. DOI 10.1016/J.Cellrep.2018.05.038. PubMed Abstract, Crossref Full Text Google Scholar. Darling, ACE 2004. Mauve, Multiple Alignment of Conserved Genomic Sequence with Rearrangements. Genome Read 14, 1394 to 1403. DOI 10.1101/.2289704. PubMed Abstract, Crossref Full Text Google Scholar. Day, R.A., 1998. How to Write and Publish Scientific Papers. Phoenix, Arizona, Oryx Press. Ding, W., Baumdicker F., and Nair, R.A., 2018. Panks, Pangenome Analysis and Exploration. Nucleic Acids Res 46, E5, DOI 10.1093/NAR/GKX977. PubMed Abstract, Crossref Full Text Google Scholar. Economo, M.N., Clack, N.G., Lavis, L.D., Gerfin, C.R., Svoboda, K., Myers, E.W. et al., 2016. A platform for brain-wide imaging and reconstruction of individual neurons. eLife 5, E10566. DOI 10.7554/eLife.10566. PubMed Abstract, Crossref Full Text Google Scholar. Ellefson, A. 2021. Towards Characterizing the Cellular 3D Proteome. Front, Bioinform 1, 598,878. DOI 10.3389/BINF.2021.598878. Crossref Full Text Google Scholar. Ivanko, D. 2010. Supplement on Visualizing Biological Data, NAT. Method 7, S1, DOI 10.1038/1Mytho310S1. PubMed Abstract, Crossref Full Text Google Scholar. Faig M., Harada R., Mori T., U. I., Takahashi K., and Sugita Y., 2015. Complete atomistic model of a bacterial cytoplasm for integrating physics, biochemistry, and systems biology. J. Mole, Graph Model 58, 1-9. DOI 10.1016/J.JMGM.2015.02.004. PubMed Abstract, Crossref Full Text Google Scholar. Farina, J., and Daggett V. 2019. Visualizing Protein Folding and Unfolding. J. Mole, Bio 431, 1540-1564. DOI 10.1016/J.JMB.2019.02.026. PubMed Abstract, Crossref Full Text Google Scholar. Gardner, A., Otan L., Barbaro B., Olson, A.J., and Goodsell, D.S., 2018. Cell Paint, Interactive Illustration of Dynamic Mesoscale Cellular Environments. IEEE Compute, Graphpal 38, 51-66. DOI 10.1109/McGee.2018.2877076. PubMed Abstract, Crossref Full Text Google Scholar. Gatto, L., Breckles, L.M., and Lilly, K.S., 2019. Assessing Subcellular Resolution in Spatial Proteomics Experiments. Kerr Oppen, ChemBio 48, 123-149. DOI 10.1016/j.cp.2018.11.015. PubMed Abstract, Cross-Ref Full Text, Google Scholar. Glenborg, N. O'Donoghue, S.I., Baliga, N.S., Gozman, A., Hibbs, M.A., Kitano, H. et al., 2010. Visualization of omics data for systems biology. NAT Method 7, S56, S68. DOI 10.1038/NMETH.1436. PubMed Abstract, Cross-Ref Full Text, Google Scholar. Ghosh, S., Matsuoka, Y., Asai, Y., Shin, K.Y., and Kitano, H. 2011. Software for Systems Biology, From Tools to Integrated Platforms. Nat Reverend Genet 12, 821-832. DOI 10.1038/Energy 3096. PubMed Abstract, Cross-Ref Full Text, Google Scholar. Gorg, C., Hunter, L., Kennedy, J., O'Donoghue, S., and Van Veek, J.J., 2013. Biological Data Visualization, Dogstool Seminar 12372. Dogstool Rep 29, 131-164. DOI 10.42/DAGREP.2.9.131. Cross-Ref Full Text. Goodsell, D.S., Franzen, M.A., and Herman, T., 2018. From Atoms to Cells, Using Mesoscale Landscapes to Construct Visual Narratives. J-Mo Bio, 430, 3954-3968. DOI 10.1016/J.JMB.2018.06.009. PubMed Abstract, Cross-Ref Full Text, Google Scholar. Goodsell, D.S., Olson, A.J., and Forley, S., 2020. Art and Science of the Cellular Mesoscale, Trends Biochem, Sci 45, 472-483. DOI 10.1016/J.Tibbs.2020.02.010. PubMed Abstract, Cross-Ref Full Text, Google Scholar. Green, E.D., Gunter, C., Biesecker, L.G., DeFrancesco, V., Easter, C.L., Feingold, E.A. et al., 2020. Strategic Vision for Improving Human Health at the Forefront of Genomics. Nature 586, 683-692. DOI 10.1038/4158602-2817-4. PubMed Abstract, Cross-Ref Full Text, Google Scholar. GTEx Consortium, 2017. Genetic Effects on Gene Expression Across Human Tissues. Nature 550, 204-213. DOI 10.1038, Nature 24277. PubMed Abstract, Cross-Ref Full Text, Google Scholar. Healy, C.G., and Innes, J.T., 2012. Attention and Visual Memory in Visualization and Computer Graphics IEEE Trans, Vis, Compute Graph 18, 1170-1188. DOI 10.1109/TVCG.2011.127. PubMed Abstract, Cross-Ref Full Text, Google Scholar. Heaven, D., 2019. Why Deep Learning AIs Are So Easy to Fool. Nature 574, 163-166. DOI 10.1038/D415860190300135. PubMed Abstract, Cross-Ref Full Text, Google Scholar. Holzinger, A., 2012. On Knowledge Discovery and Interactive Intelligent Visualization of Biomedical Data in Proceedings of the International Conference on Data Technologies and Applications Data 2012. Editors M. Helfert, C. Fancalency, and J. Philippe, Rome, Italy, Elsevier, 5-16. Google Scholar. Humphrey, W., Dalk, A., and Schulten, K., 1996. VMD, Visual Molecular Dynamics, J-Mol Graph 14, 33-38. DOI 10.1016, 0263-7855, 96, 018-5. PubMed Abstract, Cross-ref full text Google Scholar. Iwasa, J.H., 2015. Bringing Macromolecular Machinery to Life Using 3D Animation. Kerr, OpenStruct, Bio 31, 84-88. DOI, 10.1016/j.sp.2015.03.015. PubMed Abstract, Cross-ref Full Text Google Scholar. Jensen, A.B., Mosley, P.L., Oprea, T.I., Elzo, S.G., Erickson, R., Schmock, H. et al., 2014. Temporal disease trajectories condensed from population-wide registry data covering 6.2 million patients. Natcomen 5, 4022. DOI, 10.1038/5022. PubMed Abstract, Cross-ref Full Text Google Scholar. Johnson, G.T., and Herdig, S., 2014. A Guide to the Visual Analysis and Communication of Biomolecular Structural Data. Nat Reverend Mole Cell Bio 15, 690-698. DOI, 10.1038/NRM3874. Cross-ref Full Text Google Scholar. Johnson, G.T., Otan L., Alalusi, M., Goodsell, D.S., Sanner, M.F., and Olson, A.J., 2015. CellPack, a virtual mesoscope to model and visualize structural systems biology. Nat Methods 12, 85-91. DOI, 10.1038/nmyth.3204. PubMed Abstract, Cross-ref Full Text Google Scholar. Johnston, A.P.R., Ray, J., Arriety, N., Bailey, B., Lilia, A., Webb, R., et al., 2018. Journey to the Center of the Cell. Virtual Reality Immersion into Scientific Data. Traffic 19, 105-110. DOI, 10.1111/Trey.12538. PubMed Abstract, Cross-ref Full Text Google Scholar. Kerchevsky, K.J., and Snyder, M.P., 2018. Integrative Omics for Health and Disease. Nat Reverend Janet 19, 299-310. DOI, 10.1038/energy.2018.4. PubMed Abstract, Cross-ref Full Text Google Scholar. Core, S., Baldy, B., Vuong, J., and O'Donoghue, S.I., 2019. Visualization and Analysis of Epiproteome Dynamics, J. Mole, Bio 431, 1519-1539. DOI, 10.1016/J.jmb.2019.01.044. PubMed Abstract, Cross-ref Full Text Google Scholar. Core, S., Peters, T.J., Yang, P., Liu, L.D.W., Vuong, J., Crisser, J.R. et al., 2020. Temporal ordering of omics and multiomic events inferred from time series data, NPJ System Bio Opal 622 DOI, 10.1038/4154002000141-0. PubMed Abstract, Cross-ref Full Text Google Scholar. Kedrin D., Grigorievich B., Wyckoff J., Verkusha, V.V., Kandilis J., Siegel J.E. et al., 2008. Intravital Imaging of Metastatic Behavior Through a Mammary Imaging Window. Nat Methods 5, 1019-1021. DOI, 10.1038/nmyth.1269. PubMed Abstract, Cross-ref Full Text Google Scholar. Kim, S.C., Sprung, R., Chen, Y., Shu, Y., Ball, H., Pei, J., et al., 2006. Substrate and Functional Diversity of Lysine Acetylation, revealed by a proteomics survey, Mole, Cell 23, 607-618. DOI, 10.1016/J.Molecell.2006.06.026. PubMed Abstract, Cross-ref Full Text Google Scholar. Kim, J., Campbell, A.S., De Avila, B.E.F., and Wang, J., 2019. Wearable Biosensors for Healthcare Monitoring. Nat Biotechno, 37, 389-406. DOI, 10.1038/S415870190045Y. PubMed Abstract, Cross-ref full text Google Scholar. Klein, T., Otan L., Kozlakova B., Goodsell, D.S., Olson A., Groller M.E. et al., 2018. Instant Construction and Visualization of Crowded Biological Environments. IEEE Trans. Vis. CompuGraph 24, 862-872. DOI 10.1109/tvcg.2017.2744258. PubMed Abstract, Cross-Ref Full Text Google Scholar. Koch, K., McLean J., Segev R., Freed, M.A., Berry M.J., Balasubramanian V. et al., 2006. How Much the Eye Tells the Brain. Kerr, Bio, 16, 1428-1434. DOI 10.1016/j.cub.2006.05.056. PubMed Abstract, Cross-Ref Full Text Google Scholar. Kozlakova B., Krona M., Falk M., Lindo N., Baden M., Baum D. et al., 2017. Visualization of Biomolecular Structures, State-of-the-Art Revisited, Visualization of Biomolecular Structures. CompuGraph Form 36, 178-204. DOI 10.1111/cgf.13072. Cross-Ref Full Text Google Scholar. Krona M., Kozlakova B., Lindo N., Baden M., Baum D., Perulek J. et al., 2016. Visual Analysis of Biomolecular Cavities, State-of-the-Art CompuGraph, Form 35, 527-551. DOI 10.1111/cgf.12928. Cross-Ref Full Text Google Scholar. Litunik, I., and Bork P., 2019. Interactive Tree of Life, Utal, V4. Recent Updates and New Developments. Nucleic Acids Res 47, W256 W259. DOI 10.1093/NAR/GKZ239. PubMed Abstract, Cross-Ref Full Text Google Scholar. Lieberman Aiden E., Van Berkom N.L., Williams L., Immakev M., Ragus Z.T., Telling A. et al., 2009. Comprehensive Mapping of Long-Range Interactions Reveals Folding Principles of the Human Genome. Science 326, 289-293. DOI 10.1126/science.1181369. PubMed Abstract, Cross-Ref Full Text Google Scholar. Lima, M., 2011. Visual Complexity, Mapping Patterns of Information, New York, Princeton Architectural Press. DOI 10.7476, 9,788,523,209,308. Cross-Ref Full Text. Young, P., Winskog C., Person A., Lundstrom C., and Innerman A., 2006. Full-body virtual autopsies using a state-of-the-art volume rendering pipeline. IEEE Trans. Vis. CompuGraph 12, 869-876. DOI 10.1109/tvcg.2006.146. PubMed Abstract, Cross-Ref Full Text Google Scholar. Lundberg, E. and Borner, G.H.H., 2019. Spatial Proteomics, a powerful discovery tool for cell biology. Nat Reverend Mole. Cell Bio 20, 285-302. DOI 10.1038S4158001800094Y. Cross-Ref Full Text Google Scholar. Ma, DKG, Stolt C., Crisser, J.R., James D.E. and O'Donoghue S.I. 2015. Snapshot, Insulin/IGF-1 Signaling, Cell 161, 948-948.E1. DOI 10.1016/J.Cell.2015.04.041. PubMed Abstract, Cross-Ref Full Text Google Scholar. Meyer-Hein L., Vedula S.S., Spidel S., Nawab N., Kikinis R., Park A. et al., 2017. Surgical Data Science for Next-Generation Interventions, Nat Biomed Ing 1, 691 to 696, DOI 10.1038/S415510170-132-7. PubMed Abstract, Cross-Ref Full Text Google Scholar. Martinez, X, Krona M, Al-Harbi N, Rose, A.S., Laramay R.S., O'Donoghue S. et al., 2019. Molecular Graphics, Bridging Structural Biologists and Computer Scientists, Structure 27, 1617 to 1623. DOI 10.1016/J.SDR.2019.09.01. PubMed Abstract, Cross-Ref Full Text Google Scholar. Marks, V. 2021. Method of the Year, Spatially Resolved Transcriptomics, NAT. Methods 18, 9 to 14. DOI 10.1038/S41592020133Y. PubMed Abstract, Cross-Ref Full Text Google Scholar. McGill, G. 2008. Molecular Movies, Coming to a Lecture Near You. Cell 133, 1127 to 1132. DOI 10.1016/J.Cell.2008.06.013. PubMed Abstract, Cross-Ref Full Text Google Scholar. McGuffey, S.R., and Ilcock, A.H., 2010. Diffusion, Crowding and Protein Stability in a Dynamic Molecular Model of the Bacterial Cytoplasm. Employs CompuBio 6 E100694. DOI 10.1371/journal.peakb.100694. PubMed Abstract, Cross-Ref Full Text Google Scholar. Morel, W., Canis K., Kairat F., Fade, V., and Michalski, J.C., 2006. The Use of Mass Spectrometry for the Proteomic Analysis of Glycosylation. Proteomics 6, 3993 to 4015. DOI 10.1002-epemic.20600129. PubMed Abstract, Cross-Ref Full Text Google Scholar. Music, M.L., Otan L., Perulek, J., and Viola I., 2015. CellView, a tool for illustrative and multi-scale rendering of large biomolecular datasets. Eurographics Workshop, Vis Compute Biomed 2015, 61 to 70. DOI 10.2312/VCBM.20151209. Cross-Ref Full Text Google Scholar. Nayok, S., Liu H., Shu, G.I., and Iwasa, J.H. 2020, using 3D animation to visualize hypotheses. Trends Biochem, Sci 45, 633 to 634, DOI 10.1016/J.Tibbs.2020.04.09. PubMed Abstract, Cross-Ref Full Text Google Scholar. Needham, E.J., Parker, B.L., Berrykin, T., James, D.E., and Humphrey, S.J. 2019. Illuminating the Dark Phosphoproteome Psi Signal, 12, AL 8645, DOI 10.1126/Sysignal.8645. PubMed Abstract, Cross-Ref Full Text Google Scholar. O'Donoghue, S.I., Gavin, A.C., Glenborg, N., Goodsell, D.S., Herichik, J.K., Nielsen, C.B. et al., 2010a. Visualizing Biological Data, Now and in the Future. NAT Method 7, S2S4, DOI 10.1038/nmyth.f.301. Cross-Ref Full Text Google Scholar. O'Donoghue, S.I., Goodsell, D.S., Frangakis, A.S., Jossinet F., Laskovsky, R.A., Nilges M. et al., 2010b. Visualization of Macromolecular Structures, NAT Method 7, S42S55, DOI 10.1038/nmyth.1427. Cross-Ref Full Text Google Scholar. O'Donoghue, S.I., Baldy, B.F., Clark, S.J., Darling, A.E., Hogan, J.M., Core S. et al., 2018. Visualization of Biomedical Data, ANU, Reverend Biomed, Datasci 1, 275 to 304, DOI 10.1146/Anurev Biodata 080917013424. Cross-ref full text, Google Scholar. Olson, J.V., Blegaev B., Ganat F., Masick B., Kamar C., Mortensen P. et al., 2006. Global, in vivo, and site-specific phosphorylation dynamics in signaling networks. Cell 127, 635-648. DOI 10.1016/J.Cell.2006.09.026. PubMed Abstract, Cross-ref Full Text, Google Scholar. Olson, A.J., 2018. Perspectives on Structural Molecular Biology Visualization from Past to Present. J. Mol Bio, 430, 3997-4012. DOI 10.1016/J.JMB.2018.07.009. PubMed Abstract, Cross-ref Full Text, Google Scholar. U. H.D., Fawn S., Deerink, T.J., Thor A., Ellisman M.H., and O'Shea C.C. 2017. Crom-EMT, Visualizing 3D Chromatin Structure and Compaction in Interphase and Mitotic Cells. Science 357, IAG 0025. DOI 10.1126/Science.org/0025. PubMed Abstract, Cross-ref Full Text, Google Scholar. Pasoli E., Esenikar F., Manara S., Zolfo M., Karcher N., Armanini F. et al., 2019. Extensive unexplored human microbiome diversity, revealed by over 150,000 genomes from metagenomes spanning age, geography, and lifestyle. Cell 176, 649-662.E20. DOI 10.1016/J. Cell.2019.01.01. PubMed Abstract, Cross-ref Full Text, Google Scholar. Pittet, M.J., and Weiselder R. 2011. Intravital Imaging, Cell 147, 983-991. DOI 10.1016/J. Cell.2011.11.04. PubMed Abstract, Cross-ref Full Text, Google Scholar. Proctor J.B., Thompson J., Litunik I., Creevy C., Jossenet F., and Barton G.J. 2010. Visualization of Multiple Alignments, Phylogenies, and Gene Family Evolution. NAT Method 7, S16, S25, DOI 10.1038, Lesson-Meth.1434. PubMed Abstract, Cross-ref Full Text, Google Scholar. Ray, T.R., Choi J., Bhandatkar A.J., Krishnan S., Gutruf P., Tian L. et al., 2019. Biointegrated Wearable Systems, A Comprehensive Review. ChemRev 119, 5461-5533. DOI 10.1021/ox.chemrev.8B00573. PubMed Abstract, Cross-ref Full Text, Google Scholar. Riley, C., and Ingber, D.E. 2017. Art Advancing Science, Filmmaking Leads to Molecular Insights at the Nanoscale. ACS Nano 11, 12156-12166. DOI 10.1021/Axnano.7B05266. PubMed Abstract, Cross-ref Full Text, Google Scholar. Richardson, J.S., and Richardson, D.C., 1989. Principles and Patterns of Protein Conformation in Prediction of Protein Structure and the Principles of Protein Conformation. Editor G. D. Fossman, Boston, Massachusetts, Springer, U.S., 1-98. DOI 10.1007-978, 146-13-1571, 1_1. Cross-ref Full Text, Google Scholar. Richardson, J.S., 1981. The Anatomy and Taxonomy of Protein Structure. Adverb, Protein Chem 34, 167-339. DOI 10.1016/065323308, 60520-3. PubMed Abstract, Cross-ref Full Text, Google Scholar. Rosendal, J., and Harmon, L.J., 2012. 1 Zoom, a fractal explorer for the tree of life. Close Bio, 10, E10011406. DOI, 10.1371/journal.bio.1001406. PubMed Abstract, Crossref Full Text Google Scholar. Roundtree, I.A., Evans, M.E., Pan, T., and He, C., 2017. Dynamic RNA Modifications in Gene Expression Regulation, Cell 169, 1187-1200. DOI, 10.1016/J.Cell.2017.05.045. PubMed Abstract, Crossref Full Text Google Scholar. Rasavi, S.J., Beck, D.A.C., and Daggett, V. 2014. Dynamics, Data-Driven Methods and Models for Utilizing Large-Scale Protein Structure Repositories for Improving Fragment-Based Loop Prediction. Dynamics, Improving Fragment-Based Loop Prediction. Protein Side 23, 1584-1595. DOI, 10.1002/Pro.2537. PubMed Abstract, Crossref Full Text Google Scholar. Santos, A., Zafu, K., Stolt, C., Pletcher, Frankild, S., O'Donoghue, S.I., and Jensen, L.J., 2015. Comprehensive Comparison of Large-Scale Tissue Expression Datasets. Peerage 3, E1054. DOI, 10.7717/Peerage.1054. PubMed Abstract, Crossref Full Text Google Scholar. Schafferhans, A., O'Donoghue, S., and Nakamura, H., 2016. Web-based 809 molecular graphics in NI's Shonen Meeting Report, number 2016-12. Available at https://shonen.ni.ac.jp/number 2016-12.pdf. Google Scholar. Schussler, Fiorenzo, Rose, S.M., Contrapoise, K., Monaghetti, K.J., Joe, W., Mishra, T., Matarazzo, S. et al., 2019. A Longitudinal Big Data Approach for Precision Health. NataMed 25, 792-804. DOI, 10.1038/S4-159101904-14-6. PubMed Abstract, Crossref Full Text Google Scholar. Sarah, F., DiStefano, M., Spill, Y.G., Quartero, Y., Goodstadt, M., Bawu, D. et al., 2015. Restraint-based three-dimensional modeling of genomes and genomic domains. Febs Let 589, 2987-2995. DOI, 10.1016/J.Febslet.2015.05.012. PubMed Abstract, Crossref Full Text Google Scholar. Shevchenko, A., and Simons, K., 2010. Lipidomics, Coming to Grips with Lipid Diversity. Nat Reverend Mole, Cell Biol 11, 593-598. DOI, 10.1038/NRM2934. PubMed Abstract, Crossref Full Text Google Scholar. Singler, J., McClary, K.M., White, K.L., Albert, F., Solly, A., and Stevens, R.C., 2018. Opportunities and Challenges in Building a Spatiotemporal, Multi-Scale Model of the Human Pancreatic Beta Cell, Cell 173, 11-19. DOI, 10.1016/J.Cell.2018.03.014. PubMed Abstract, Crossref Full Text Google Scholar. Summer, B., Baden, M., Krona, M., and Woods, A., 2018. From Virtual Reality to Immersive Analytics in Bioinformatics. J. Integer Bioinforma, 15, 20,180,043. DOI, 10.1515/Jib 2018-0043. PubMed Abstract, Crossref Full Text Google Scholar. Stahl, P.L., Salmin, F., Vikovic, S., Lundmark, A., Navarro, J.F., Magnuson, J., et al., 2016. Visualization and Analysis of Gene Expression in Tissue Sections by Spatial Transcriptomics. Science 353, 78-82. DOI 10.1126 Science.off 2403. PubMed Abstract, Cross-Ref Full Text Google Scholar. Stickles, R.R., Murray, E., Kamar, P., Lee, J., Marshall, J.L., DiBella, D.J. et al. 2020. Highly sensitive spatial transcriptomics at near cellular resolution with slide-seq 2 Nat Biotechnology 39, 313-319. DOI 10.1038/4158702007339-1. PubMed Abstract, Cross-Ref Full Text Google Scholar. Tomita, M. 2001. Whole Cell Simulation, A Grand Challenge of the 21st Century. Trends Biotechnology 19, 205-210. DOI 10.1016/SO1677799, 01, 16365. PubMed Abstract, Cross-Ref Full Text Google Scholar. Ulin, M., Zhang C., Lee S., Huachet, E., Fagerberg L., by Corey G. et al., 2017. A Pathology Atlas of the Human Cancer Transcriptome. Science 357, Eon 2507, DOI 10.1126/Science.on 2507. PubMed Abstract, Cross-Ref Full Text Google Scholar. Valdez-Mora F., Salomon R., Gloss B., Law, AMK, Castillo L., Murphy K.J. et al., 2019. Single-cell transcriptomics reveals involution mimicry during the specification of the basal breast cancer subtype. Cell Rep 35, 108,945, DOI 10.1101 624,890. Cross-Ref Full Text Google Scholar. Vander Motten L. and Hinton G. 2008. Visualizing Data Using TSNE JMOC, Learn Re 9, 2579-2605. Google Scholar. Vandenbaan A. and DSD, 2020. A clustering independent method for finding differentially expressed genes in single-cell transcriptome data. Nat Command 11, 4318. DOI 10.1038/S4146702-01790003. PubMed Abstract, Cross-Ref Full Text Google Scholar. Walden, N., Waldner, M., LeMusic, M., Groller, E., Goodsell, D.S., Otan L. et al., 2019. Cuttlefish, Color Mapping for Dynamic Multi-Scale Visualizations. Compute Graph, Form 38, 150-164. DOI 10.1111/CGF.13611. PubMed Abstract, Cross-Ref Full Text Google Scholar. Walter T., Shattuck, D.W., Baldick R., Baston, M.E., Carpenter, A.E., Ducci S. et al., 2010. Visualization of image data from cells to organisms. Nat Method 7, S26S41. DOI 10.1038/Myth.1431. PubMed Abstract, Cross-Ref Full Text Google Scholar. Waterhouse A.M., Proctor J.B., Martin D.M.A., Clamp M., and Barton G.J., 2009. JAVU version 2, a multiple sequence alignment editor and analysis workbench. Bioinformatics 25, 1189-1191. DOI 10.1093 Bioinformatics/BTP033. PubMed Abstract, Cross-Ref Full Text Google Scholar. Watson, J.D., and Crick, F.H.C., 1953. Molecular Structure of Nucleic Acids. A Structure for Deoxyribose Nucleic Acid. Nature 171, 737-738. DOI 10.1038/171737A0. PubMed Abstract, Cross-Ref Full Text Google Scholar. Wickham, H. 2009. ggplot 2. Elegant Graphics for Data Analysis. Use R. New York, Springer, New York. Google Scholar. Wills, Q.F., Livak, K.J., Tipping, A.J., Inver T., Goldson, A.J., Sexton, D.W. et al., 2013. Single-cell gene expression analysis reveals genetic associations masked in whole tissue experiments. Nat Biotechno 31, 748-752. DOI 10.1038/NBT.2642. PubMed Abstract, Cross-Ref Full Text Google Scholar. U. K.H. and Kahan, I.S., 2019. Framing the Challenges of Artificial Intelligence in Medicine. BMJ Qualsoft 28, 238-241. DOI 10.1136/BMJQS 2018-008-551. PubMed Abstract, Cross-Ref Full Text Google Scholar. Zheng, Y., Wang, X., and Kelleher, N.L., 2016. Epiproteomics, Quantitative Analysis of Histone Marks and Codes by Mass Spectrometry. Kerr, Oppen, Chem Bio 33, 142-150. DOI 10.1016/J.Kupa.2016.06.007. PubMed Abstract, Cross-Ref Full Text Google Scholar. Keywords: Bioinformatics, Data Science, Data Visualization, Visual Analytics, Computational Biology, Science Communication. Citation: O'Donoghue S.I. 2021, Grand Challenges in Bioinformatics Data Visualization, Front, Bioinform, 1-669,186, DOI, 10.3389/FBINF.2021.669186. Received, February 18, 2021. Accepted, April 30, 2021. Published, June 17, 2021. Edited by Barbora Kozlakova, Masaryk University, Czechia. Reviewed by Pierre Pau Vasquez, Universitat Politècnica de Catalunya, Spain. Copyright 2021 O'Donoghue. This is an open-access article distributed under the terms of the Creative Commons Attribution License, CC BY. The use, distribution, or reproduction in other forms is permitted, provided the original authors and the copyright owners are credited, and that the original publication in this journal is cited in accordance with accepted academic practice. No use, distribution, or reproduction is permitted, which does not comply with these terms. Correspondence: Sean I. O'Donoghue, sean@o'donoghulab.org. Disclaimer: all claims expressed in this article are solely those of the authors and do not necessarily represent those of their affiliated organizations, or those of the publisher, the editors, and the reviewers. Any product that may be evaluated in this article or claim that may be made by its manufacturer is not guaranteed or endorsed by the publisher. Specialty Grand Challenge article. Front, Bioinform, June 17, 2021. Secretary Data Visualization. Volume 1-2021, https://doi.org/10.3389/FBINF.2021.669186. Grand Challenges in Bioinformatics Data Visualization. www.frontierson.org. Sean I. O''Donoghue 1,2,3. 1 Garvan Institute of Medical Research, Darlinghurst, NSW, Australia. 2 School of Biotechnology and Biomolecular Sciences, University of New South Wales, Kensington, NSW, Australia. 30 Data 61, Eveleigh, NSW, Australia. Introduction. Increasingly, the life sciences rely on data science, an emerging discipline in which visualization plays a critical role. Visualization is particularly important with challenging data from cutting-edge experimental techniques, such as 3D genomics, spatial transcriptomics, 3D proteomics, epiproteomics, high-throughput imaging, and metagenomics. Data visualization also plays an increasing role in how research is communicated. Some scientists still think of data visualization as optional. However, as more realize it is an essential tool for revealing insights buried in complex data, bioinformatics visualization is emerging as a subdiscipline. This article outlines current and future grand challenges in bioinformatics data visualization and announces the first publication venue dedicated to this subdiscipline. Over the past 2 decades, life science data have increased rapidly in volume and complexity, with the result that data analysis is often the major bottleneck. O'Donoghue et al., 2010a. For example, all major genomics breakthroughs so far have been accompanied by the development of groundbreaking statistical and computational methods. Green et al., 2020. Thus, in the remaining decades of the 21st century, life scientists will become increasingly reliant on the emerging tools and methods of data science. Bly and Smith, 2017. Altman and Levitt, 2018. One of these methods is data visualization, aka data vis, which plays a critical role in transforming data and analysis outcomes into insight. Card et al., 1999. Data visualization involves analysis, design, and rendering, as well as observation and cognitive processing. Figure 1. Some scientists think of data vis as an optional step mostly aimed at aesthetics. However, there is growing recognition that it is an essential tool in the analysis of complex data. Two indicators of this recognition are the recent sales of data vis companies Looker and Tableau for UseDeb and $16 billion respectively. Figure 1. www.frontierson.org. Figure 1. The data science cycle. Analysis of newly acquired data increasingly relies on integration with large accumulating volumes of complex pre-existing data and requires frequent reanalysis and re-rendering. Visualization is the main way researchers observe both raw and analyzed data. An overarching grand challenge of data visualization is to leverage human visual capabilities, which involve most of the brain and can process 10 million bits per second, Koch et al., 2006, to recognize patterns within 100 milliseconds, Healy and Enns, 2012, thereby transforming data into insight. These insights, in turn, lead to new hypotheses, thus continuing the cycle. Unfortunately, the critical step of manually validating derived models by visually comparing raw versus analyzed data, Anscombe 1973, is often overlooked. Currently, however, most attention is focused on another aspect of data science, namely, the use of machine learning to develop artificial intelligence systems. Such systems have recently led to exciting advances in the life sciences, for example, Callaway 2020a, but also to some hyperbole. Clearly, machine learning methods are increasingly critical for research, but these methods also have limitations. Challen et al. 2019, Heaven 2019, Yu and Kahan 2019. More fundamentally, automated methods are insufficient, since analysis outcomes must be observed and understood by an analyst before insight can occur. Figure 1. Most analysts use data visualization as an integral part of their cognitive processes. Especially important is manual validation, which involves checking for errors and outliers in raw data, and for wrong assumptions used in automated analysis methods. Anscombe 1973. Automated data